Good afternoon, learners. Welcome you all back to the Gyan Darshan session. Today we will be taking up one of the courses, that is Lab Course 2. And this is a laboratory course which you have in your second semester in the MSc Zoology program. Now we will take up 4 experiments for this Lab Course 2. We will discuss today 4 experiments. That is experiments 1, 3, 4 and 5. So now take a look at what are the topics for these experiments. We will come one by one. The first experiment that you have is the genomic DNA isolation. Now first of all we should know that what is meant by the genomic DNA and why do you need to isolate genomic DNA? Now, you know that any organism, right? Like when we are talking of the microbes or the plants or the animals, we have the genome, and within the genome you have the chromosomal DNA. So we need to learn how to isolate the DNA. Why? Because, for instance, you want to sequence. You want to know the sequence of the DNA. So you need to isolate the DNA first and then use that DNA as a template for sequencing. Or let's say you are doing some genetic engineering. You are cloning a particular gene. Okay? And then you are integrating the gene within the genome of a plant or an animal. So you need to know whether the gene has been incorporated within the genome. So for that, what you have to do? You have to again isolate the genomic DNA. So for all these purposes, there are many other reasons. But these are some of the prime reasons for which you have to know how to isolate the genomic DNA. So because this is a practical session, or the today's discussion is mostly based on the laboratory experiments, so we will focus our attention mostly on the principles, and then we will take a look at the procedure, that is how do you go for this kind of work, what are the protocol steps for isolation, and then the observation, and finally the precautions. When you are doing the experiments, what precautionary measures that you should take so that you get the best quality, or you get the best result, or you get the best observation. So those will be discussed in today's discussion based on these 4 experiments as we have highlighted. So I think it is clear that why we need to isolate the genomic DNA. Now this, as I said, that you can isolate the genomic DNA from bacteria, from plant and animal. Now we will focus our attention on the genomic DNA isolation from bacteria. So the DNA can be extracted from bacteria as a high molecular weight polymer of deoxyribonucleotides via enzyme or alkaline method. So which is the method that we are using? We are using the enzyme or the alkaline method. Now what is the main principle? Now when you are isolating the genomic DNA in any system, whether in bacteria, plant or animal, we normally use one detergent. This detergent, it has the name CTAB in abbreviation, that is cetyl trimethyl ammonium bromide. This is a cationic detergent. So your lab should have this chemical. Now when you are using this CTAB in solutions of high ionic strength, CTAB forms complexes with proteins and most of the polysaccharides, but does not precipitate nucleic acids. Because when you are isolating genomic DNA, you all know that along with DNA, you have the different proteins, associated proteins, and also the polysaccharides. Okay, so you know about the histones, and there are also the non-histone proteins which are associated with DNA. So you need to remove this. You need to remove the proteins. You need to remove the polysaccharides. For that, what you do is use this cationic detergent, because this CTAB, it forms complexes with proteins and polysaccharide but does not precipitate nucleic acid. And sometimes what we do is we add another agent called polyvinyl pyrrolidone, that is PVP, and high salt solution to get rid of phenolics and polysaccharides from the isolated genomic DNA, particularly when you are isolating genomic DNA from plant tissue. Okay, because in plants you have a lot of polysaccharide, you have the accumulated starch. Then you also have the different secondary metabolites like phenolics, which interfere with the quality of the genomic DNA. So these need to be removed. So for that, all these purposes, you use this PVP, that is polyvinyl pyrrolidone. So when you are preparing the extraction buffer, make sure that it should have the CTAB together with the PVP. So you have to prepare the extraction buffer containing these particular chemicals, that is cationic detergent, that is CTAB, and together with the PVP. So what happens is CTAB polysaccharide protein complexes, these are then removed, and you have to, of course, undergo a centrifugation process in a centrifuge machine, and then extract with phenol and chloroform, and you get the genomic DNA in the purified form after precipitation with chilled ethanol. That is absolute ethanol, which should be kept under cold condition. You can add that ethanol and you can precipitate the nucleic acid. Alternately, you can also use isopropanol for precipitating the DNA or the nucleic acids. Now, you can do this, you can follow this protocol for the bacteria also. That is because we are today mostly talking about the isolation of genomic DNA from the bacterial cells. So what are the materials required for this experiment? First, as I said, CTAB 10%, pre-warm before use. Then you need the absolute ethanol, ice cold. You need 70% ethanol. Then you want to check the quality of the DNA. For that, you need agarose. If you remember, in one of my earlier laboratory sessions, I discussed on some of the experiments like plasmid DNA isolation, agarose gel electrophoresis, etc. So today I will not be repeating the same experiments. You can follow my earlier discussion, which is available in the YouTube. You can just observe that and you can get the idea if you have not followed the earlier practical sessions. So agarose, ethidium bromide, then RNase A, 10 mg per ml, properly boiled to remove DNase1, water, microfuge tubes, microtips, micropipette. So these are some of the materials that you required, and in addition to that, some of the buffers like tris EDTA pH 8. You need sodium chloride, 5 molar sodium chloride. Then you need the CTAB buffer. How will you prepare the CTAB buffer? That is 10 gram CTAB in 100 ml distilled water. And then you have to autoclave it. That means it is 10% CTAB because 10 gram in 100 ml. And you need chloroform isoamyl mixture, that is 24:1 ratio, equilibrated with a layer of TE buffer pH 8. Phenol chloroform isoamyl alcohol mixture, that is 25:24:1, that is volume ratio, equilibrated with a layer of TE buffer pH 8. And then sodium acetate, 3 molar sodium acetate pH 5.2, and then you have to autoclave it. So all the chemicals or all the buffers that you use, they should be properly prepared and they should be autoclaved. Okay, without autoclaving you cannot do, because otherwise the DNA will be degraded. So you have to prepare all the buffers, you have to autoclave the buffers, and then, or any chemicals, even the glasswares and the plasticwares that you are using, all the chemicals need to be autoclaved. Even the water, not only double distilled water, but the water also has to be autoclaved, sterile, and then you can perform the experiment. Now the procedure. So first what you have to do is you have to grow the bacteria, because you are isolating it from bacterial cell. Grow the bacterial cell in 5 ml of nutrient broth. Pellet the cells by centrifugation at 5000 revolutions per minute for 10 minutes. So you are actually pelleting the bacteria, and then you are discarding the supernatant. So all the bacteria is now pelleted down. Next, what you do, 400 microliter of TE, that is tris EDTA pH 8, and 100 microliter NaCl, that is 5 molar NaCl stock, you add to the pellet, resuspend the cells gently. So you have to tap the cells and you have to dissolve the cells within this buffer. Then you add the lysozyme. Lysozyme, what is lysozyme enzyme? It is an enzyme. What is the function of the lysozyme? The lysozyme enzyme is used to degrade the cell wall, because you know that the bacteria, they have the cell wall. You have the peptidoglycan layer. Okay, and if the genomic DNA is to be isolated, then you have to degrade the cell wall. Otherwise, how can you isolate? So even for a plant cell which has the cell wall, sometimes you need to have some harsh treatment. There we don't add the lysozyme, but there we add some other agents. But for bacterial cell, you have to use the lysozyme. You need to use the lysozyme, particularly if it is a gram-negative bacteria, which has a very thick cell wall, like Escherichia coli, which has a very thick cell wall. Okay, so there you need this lysozyme enzyme. Then for 1 hour at 37 degrees centigrade water bath, you have to incubate the mixture. Then you add proteinase K. Why proteinase K? Because proteinase K is the enzyme that will degrade all the proteins, because you don't need the protein, you need only the DNA part. So you can add proteinase K. Again, you incubate for 1 to 16 hours at 60 degrees centigrade. Finally, you add the CTAP buffer. That is 10% CTAP buffer, which you have already prepared. So you add 100 microliter of CTAP, mix, incubate at 65 degrees centigrade for 1 hour by occasional inversion of the tube. Then you add RNase A. Why you are adding RNase A? Because you don't want RNA. You want to avoid the RNA contamination. So you are adding RNase A. That is another enzyme which will degrade the RNA, but not the DNA. Next, you have to add phenol chloroform isoamyl alcohol 500 microliter, mix it thoroughly and incubate on ice for 30 minutes. And finally, you have to centrifuge. Don't go for very high speed in the centrifuge machine. Why? Because genomic DNA or chromosomal DNA is a high molecular weight. It has very high molecular weight. So if you use very high speed, then the chances are that the DNA might get degraded. So the speed should, in any case, the speed should never exceed 10,000 revolutions per minute. If possible, you can keep it a bit low also, like 8,000 revolutions per minute also you can use. So the centrifugation is done for 10 minutes. Then what you do is you collect the supernatant. Don't discard the lower portion which has all the cell debris and all. The proteins, again for plant cell, if it is a plant cell, then polysaccharides. In bacterial cell, mostly the proteins. So you avoid that layer. You take the clear supernatant from above in a fresh tube. And then you have to add chloroform isoamyl alcohol, that is 500 microliter. Again you mix it. And then don't mix it vigorously for the same reason that the DNA might get degraded. You have to handle it very gently. And then centrifuge it for 10 minutes. Collect the supernatant, and now you have to precipitate the DNA. How will you precipitate it? For that you have to use two agents, that is sodium acetate pH 5.2, 1/10 volume of sodium acetate pH 5.2 and 2 volumes of ice cold ethanol to precipitate the DNA. And for that you have to keep the tube, microfuge tube, at -20 degrees centigrade for 1 hour. If necessary, sometimes we can incubate it overnight also. That is -20 degrees centigrade overnight. Or minimum, it is preferable 2.5 hours, 2 to 2.5 hours is preferable. For E. coli, okay, fine, you can, I mean, for bacteria or E. coli cell, you can incubate for 1 hour also, but preferably sometimes you need to increase the time, like 2.5 hours, 2 to 2.5 hours, or sometimes maybe overnight also. Next, what you have to do, again you have to centrifuge it down. So, but cold condition, that is at 4 degrees centigrade. So, in your centrifuge machine, you should have a refrigerated system where you can undergo the cooling process, and you can set the temperature to 4 degrees centigrade in a centrifuge machine and then run the centrifugation for 10 minutes. Now what you will do? You have to retain the pellet, because now the DNA is pelleted down, and you have to wash it with 70% ethanol, chilled 70% ethanol. Why do you need this washing? Because you need to remove the salt, associated salt mostly. So allow to stand for 5 minutes, and then maybe again you have to undergo a centrifugation, drain off the excess ethanol and semi-dry the pellet. And then finally dissolve the DNA pellet in 100 microliter 3 CDTA pH 8. So gentle heating may be required to dissolve the pellet completely. And finally, you have to store the DNA, dissolved DNA at minus 20 degrees centigrade. And then you have to finally check the quality of the DNA by running the DNA in 0.8% to 1% agarose gel, that is through agarose gel electrophoresis. What will be your observation? The observation will be like this, as shown here. That is, you have a sharp band, as you can see. In lane number 2 and lane number 3, you can see sharp band. These are the DNA bands. So if you have isolated your genomic DNA or chromosomal DNA in a proper manner, then the figure should come like this. That is a sharp, distinct band. But if your DNA is degraded, then you will get a smear. Okay, you will get, that means the DNA has been fragmented, or the DNA has been broken into small fragments. Okay, and therefore you will not get this sharp, intense band, intact band. Okay, so that is how, this is the ideal picture which is expected for an intact genomic DNA. Now, what are the precautions that you need to take for these experiments? Now, CTAP should be handled carefully. Because it is a strong detergent, it can damage eyes and irritate your skin. Chloroform, phenol, both of them should be handled very carefully. Chloroform is toxic by inhalation and it can damage the eyes. Phenol, it is toxic and corrosive to the skin and eyes. All the buffers, water, glassware, plasticware, I think I have already mentioned this, that they should be properly autoclaved. They should be sterile. Otherwise, if there is DNase contamination, then your entire DNA will be fragmented. Okay, you will not get the intact band. So the autoclave part is very important, and you have to maintain the sterile condition of all the reagents, buffers, chemicals, and the glasswares and plasticwares. Now we come to the next experiment, that is your experiment number 3, that is polymerase chain reaction. In short, it is called PCR. Now, perhaps by this time, who are the students who have started your courses, either in semester in the first year or in the second year, by this time you should be familiar with what is meant by PCR. Polymerase chain reaction. What is the purpose of doing PCR? That is, you want to know whether one particular gene is present in the genome. Okay. So for that, what you have to do? You have to do this PCR experiment. Take any gene, okay, actin gene, for example. Actin, is a cytoskeletal protein. And the actin gene, whether actin, it is a constitutive gene, that is, which is present in all the systems. Now, if you want to know whether actin gene is present in the genome, what you will do? You have to do this PCR experiment. So before coming into the technical part, we should just know the name of the person who actually identified or who first actually described this PCR. That is Kary Mullis. He first described this amplification okay, of a single copy mammalian gene. Like one gene is present, it is present in a very low copy. Now you want to amplify it. How will you amplify it? You can use this polymerase chain reaction. So basically it is also a technique for DNA amplification. So the enzyme which is used for this purpose for undergoing a PCR reaction or polymerase chain reaction is known as Taq polymerase. Taq stands for Thermus aquaticus. So this is one of the bacteria, okay, thermophilic bacteria, from where the enzyme was first isolated. That is Taq polymerase. It is a heat resistant DNA polymerase which is used for DNA amplification. Okay. Now, what is the basic principle of PCR? If we now quickly look at it, or rather, what are the things that you need for undergoing a PCR reaction? So, first of all, you need a pair of synthetic oligonucleotide primers, and these primers are designated as forward and reverse primer. That is, if you know the sequence of the gene, then from the 5' end, you have to design one primer, and from the 3' end, you have to design another primer. From the 5' end to 3' end, okay, the primer that you are designing, that is the forward primer, and the 3' to 5' end, that from the 3' end, the primer which you are designing, it is the reverse primer. So you have to check the sequence of the gene, and accordingly you have to prepare the forward primer and the reverse primer. This is first step. And these two primers should not differ too much in length. That is, each of them should be around 20 to 30 nucleotides in length. 20 nucleotide, you can say the minimum length, and it's preferable not to go beyond 30 nucleotides. And they should have a balanced content of GC residues and less propensity of secondary structure formation that help in the priming DNA synthesis. So the primer should be such. So what are the important criteria of primers? Synthesizing primers or designing primer. Number one, their length, and number two is what is their nucleotide composition. Okay, these two things have to be kept in mind in addition to the propensity of the secondary structure formation. Now you take the template DNA. Template DNA means the DNA which you will use for amplifying the gene. So that template DNA, it can be a plasmid DNA or it can be a genomic DNA. Okay. Then what you need is the divalent cation magnesium. Normally we use magnesium chloride. Why is it required? It is required for the proper functioning of the thermostable DNA polymerase. And normally we add 1.5 millimolar magnesium chloride to the reaction buffer. Then what you need? You need the DNTPs, deoxynucleoside triphosphate. That is, there are 4 DNTPs, D-ATP, D-TTP, D-CTP and D-GTP. Because you know, adenine, thymine, cytosine and guanine. So accordingly, you have the 4 DNTPs and usually 200 to 250 micromolar of each. So these are the major components, and the working concentration of the DNTPs, mind that they should not exceed greater than 4 millimolar, because otherwise they might hinder the polymerase chain reaction because they will sequester the magnesium ions. And sometimes what happens is in this kind of reaction, you need to standardize the concentration of magnesium chloride and DNTPs. What is the concentration that you are going to add? Sometimes you have to, when you are doing experiments, you have to standardize it through different, trial and error. Sometimes, though there is a recommended concentration, but sometimes we need to adjust it depending on the nature of the gene. So those kind of things, if at all in future, if you are pursuing your research, then you might need to, standardize those kind of composition, okay, or constituents through trial and error. The reaction is carried out in presence of 10 millimolar tris chloride buffer adjusted to pH between 8.3 and 8.8. And as I said, the Taq polymerase enzyme that is isolated from thermophilic archaebacteria called Thermus aquaticus. So you have to add the Taq polymerase also at 0.5 to 2.5 units per 25 to 50 microliter reaction. So that is now coming to the steps. What are the steps in a PCR? So we have seen that what are the components. Now we take a look at what are the steps. There are mainly 3 steps. First is denaturation. What is denaturation? Denaturation of the template DNA means you have to denature the template, because you know that DNA is a double-stranded structure, double helical structure. So you need to denature the DNA. That means the double helical structure needs to be, obstructed. It needs to be hindered. So how do you denature it? Through applying high heat. Okay. Then second step is annealing. What is annealing? Annealing means now the oligonucleotide primers will bind, or the oligonucleotide primers will anneal to the single-stranded target sequences. Third point, extension of the anneal primers by Taq polymerase. That means now the extension is occurring. So denaturation, annealing and extension. How do you carry denaturation? High temperature, 94 to 95 degrees centigrade for 45 seconds. Sometimes it might be extended to 1 minute also. Then primer annealing. What is the temperature that you need to set for this primer annealing? That is 3 to 5 degrees centigrade lower than the calculated melting temperature at which the dissociation of primers from their template occurs. What is melting temperature? What is the formula for calculating the melting temperature? That is, you have to calculate based on the primers. Twice 80 plus 4 GC. This is the formula. 80 means A plus T, GC means G plus C. Twice 80 plus 4 GC, remember this. So based on that you can calculate the melting temperature or TM of the primer. So for forward primer you will have 1 TM, and for reverse primer you will have another TM. And plus minus 5 can be adjusted to set the final annealing temperature. Next, extension. Extension is carried out at 72 to 78 degrees centigrade for 1 minute for every 1000 base pair of the product. Polymerization rate of Taq polymerase is 2000 nucleotides per minute at this temperature range. Normally, how many cycles do we need to carry? 25 to 30 cycles. It is recommended 30 cycles. Sometimes we may go up to 35 cycles. But it is preferable not to go beyond 35 cycles. So ideally 30, and in extreme cases, if you need more amplification, then 35. But after that, it becomes saturated. There is no point in increasing the number of cycles to increase the copy number of the DNA or the copy number of the gene or the sequence, whatever. So materials required, if we take a quick look, thermal cycler, microfuge tubes, micropipette. Of course, again you have to carry out the agarose gel electrophoresis, which is common because you have to run the gel and see if the DNA has been amplified or the gene has been amplified or not. Buffers and solutions, 10x PCR amplification buffer, DNTP mix solution containing all the 4 DNTPs, Taq polymerase. So you see the procedure. So in a tube, you have to set the following reaction. You have to take a buffer, that is PCR amplification buffer, because you know that all enzymes require buffer. So you have to use buffer. From 10x stock solution, you have to make it 1x. Magnesium chloride, solution of 4 DNTPs, forward primer, reverse primer, Taq DNA polymerase, and water for adjustment. And of course the template DNA. So you can make the final volume to 50 microliter. Accordingly you can calculate and you can add in the reaction. And always there should be a positive control. In fact, for any experiment, you need to have a positive and a negative control. So positive control will determine the efficiency of PCR. Negative control, why negative control? Because that will detect the contamination. You have to ensure that there are no contaminations. So for this PCR, you need a particular machine that is called thermal cycler. And whatever we have discussed, that the setting up of the PCR cycle, as you can see, you have to program the machine. You have to program the thermal cycler. So cycle number 30 cycle, denaturation step 94 degrees, say 30 seconds, annealing, again let's say 55 is the annealing temperature or TM that you have calculated. So 30 seconds, it may extend to 1 minute. Polymerization 72 degrees centigrade 1 minute. Last cycle, that is 1 minute at 94, 30 seconds at 55 and 1 minute at 72 degrees centigrade. So this is how you can, this is a rough format. Of course, you might have to change. I mean, you might have to, as I said, do trial and error and set the number of, set the temperature as well as the time. Okay. This might vary depending on the reaction, which you might need to adjust during your course of the experiment. And then you load the reaction in an agarose gel. So you need to observe, okay, whether you have got the amplified DNA or not. And this is actually the picture. So if you get a proper PCR product, as you can see in lane number 4, 5 and 6, okay, in lane number 2 and 3, you see these are negative control. So you are not getting any band, okay. And lane number 1, that is the molecular weight marker against which you can check the size of the amplified product, whether you have got the proper amplified product or not, because each gene, it has a particular length, base pair. So how will you understand that whether the proper gene has been amplified or not? It should have a specific molecular weight or base pair, which you can compare against the standard molecular weight marker, which you have loaded in lane number 1. So that is how your experiment is complete. Again, what are the precautions? All the glasswares, plasticwares, reagents, they should be autoclaved. Then Taq polymerase is an enzyme. You have to properly store it at minus 20 degrees centigrade. Place all the components on ice bath during preparation of reaction mixture. Add the enzyme at the end. Add Taq polymerase at the end. Don't add it at the beginning, because once the enzyme is added, the reaction will start. So once you are ready with everything, finally add the enzyme and quickly put it in the thermal cycler machine. Don't wait much. Mix the reaction gently. Calculate the melting temperature of the 2 primers. I told you the formula, twice AT plus 4 GC. This is the formula by which you can calculate. And do not exceed 35 cycles. Try to keep it at 30, max to max 35. So that's the points which you should consider while, when you are doing the PCR reaction. So this is a very important experiment for molecular biology. Isolation of genomic DNA, doing the PCR, these are very common experiments. Now, once we know how to isolate DNA, we should also know how to isolate a protein and how to estimate the protein content. Qualitative and quantitative. So today's second part of the discussion, the next 2 experiments, these will focus on protein isolation and qualitative and quantitative detection of the protein quality. That means the quality of the protein is being checked and the quantity of the protein is being measured. Because we know that we are surviving because we have the proteins. Any organism, whether bacteria, plant or animal, these proteins are actually building blocks. So there are several such proteins within the body of an organism, and some of which are expressed continuously and some of which are expressed in response to a particular situation. whether constitutive or inducible, whatever, we need to isolate the protein. We need to isolate the total protein content. So for that we have a protocol. We have, in fact, from what is the source from where you are isolating the protein. That is important. Now, before going to isolation, if I focus on the estimation part, then there is a method which is called the Lowry method, by which you can estimate the protein content. What is the principle of Lowry method? Hydrolyze the protein and then estimate the amino acid. So it's a very sensitive method where you use one reagent called Folin CO-Cal2 reagent. And this reagent, it has phosphomolybdic phosphotungstic component. So when you add this reagent, what happens is it interacts with the amino acids tyrosine and tryptophan, which is present in the protein, and then a blue color is generated, and this blue color is developed by the biuret reaction of the protein with the alkaline cupric tartarate, which is measured in the Lowry's method. So basically you are using this reagent, Folin CO-Cal2 reagent, and the alkaline cupric tartarate. So this particular method, it is applicable for the buffer soluble proteins, that is soluble proteins. You cannot measure the aggregated proteins or the hydrophobic proteins through this method. Only the proteins which go to the soluble phase, only those proteins can be detected using Lowry method, and this is very useful method. Like any lab which is working on biochemistry or the protein chemistry, or let's say molecular biology, so any lab which is working on these areas, they should know how to do this method, or how to estimate the protein through Lowry method. Now we come to the isolation part, that is how you will isolate the protein. Because only you can estimate the protein when you learn to isolate it, like we learned the isolation of genomic DNA. So for that you need isolation buffer, that is tris chloride pH 7.5, magnesium chloride, potassium chloride, sucrose, PMSF, phenyl methane sulfonyl fluoride, which is a protease inhibitor, and 1 millimolar DTT dithiotritol. This is also a protease inhibitor. That means which inhibits the protease. And what does protease do? Protease degrade the protein. So these reagents, they help in protein stabilization. Then you have the phosphate buffer saline, or PBS, which is pH 7.4. So you can use this, that is, it's a solution containing disodium hydrogen phosphate, sodium chloride, potassium chloride, and potassium dihydrogen phosphate. Then lysis buffer, that is, you mostly have tris chloride buffer, EDTA, as the chelating agent, and very importantly you have sodium dodecyl sulfate, which is a detergent, 0.5% SDS you have to use in the lysis buffer. Now for protein estimation part, you need spectrophotometer, cuvette, 2% sodium carbonate in 0.1 normal sodium hydroxide, which you designate as reagent A, 0.5% copper sulfate in 1% potassium sodium tartrate, reagent B, alkaline copper solution you have to prepare, which is reagent C, by mixing A and B in proper ratio. So basically you need this reagent C for the protein estimation. And as we have mentioned that Folin CO-Cal2 reagent, that is reagent D, which contains sodium tungstate, sodium molybdate, phosphoric acid, and concentrated hydrochloric acid. So the reactions are given here. When you prepare the solution, you can prepare it in this fashion. And of course the protein stock. Like if let's say if you take any, if you want to estimate the protein, and if you learn how to do this technique or how to perform this technique, you can use any standard protein like bovine serum albumin, BSA, we call it BSA. So you can use this BSA, you can use a stock solution of BSA, and from the stock solution you can prepare the gradually increasing concentration of BSA and then learn how to estimate it. Okay. So procedure. Now, how will you isolate the protein? It's pretty simple. You have already prepared the extraction buffer. What you do is you homogenize the sample in the buffer, centrifuge it, okay, and then collect the supernatant. So now your supernatant will actually have the all the proteins, because it's a buffer soluble protein, as I said. Because we are not learning about the aggregated proteins. We are only learning about the buffer soluble proteins, that is, which go in the soluble phase. Or let's say if you are extraction of the proteins, if you are extracting proteins from the cells in suspension, that is suspension cultured cells, then you have to again centrifuge the cell suspension, discard the supernatant, get the pellet, then add ice cold phosphate buffer saline, again go for centrifugation, then you take the pellet, add ice cold lysis buffer to the cell pellet, again centrifuge it, collect the supernatant in fresh tube. Now you need to discard the pellet because you have undergone cell lysis. So it will go to the supernatant. So now use this supernatant as a source for the total protein. Or let's say if you are extracting the proteins from tissues, then you have to dissect the tissue, and again same procedure, that is the homogenization buffer and the lysis buffer you have to add, centrifuge it and collect the supernatant, discard the pellet. Now protein estimation, because now the protein that you have isolated, it's an unknown protein. So if you want to know the concentration of the total protein in your sample, for that you need a standard. What would be the standard? The standard would be the bovine serum albumin. So first what you have to do is you have to prepare, the gradually increasing concentrations of bovine serum albumin and prepare a standard curve based on the bovine serum albumin estimation, and then what is the protein content in your sample that has to be compared with respect to this standard that you have prepared with the gradually increasing concentrations of the BSA. So what you do is you prepare a stock solution of BSA, and then you can follow this particular table. Like here you see that you have the protein standard, that is BSA. You prepare the gradually increasing concentration in different tubes, 0.1 ml, 0.2 ml, 0.3 ml up to 0.9. Okay, and then you add the alkaline copper solution and Fol-in-Cal-2 reagent. So you will, as I discussed, you will get a blue color generated. So that blue color you can, estimate the intensity of the color in a spectrophotometer. So you can take the readings, and based on that readings you can prepare a standard curve, and against that standard curve you can similarly undergo a reaction with the unknown protein that you have isolated and compare the concentration of that protein with respect to this standard that you have prepared using bovine serum albumin or BSA, using this kind of reaction. So that is the way by which you can estimate the protein. So you have to check the quality of the isolated protein by visualization of protein bands through sodium dodecyl sulfate polyacrylamide gel electrophoresis, which will be our next experiment and the last experiment for today, which we will discuss. So that is the qualitative part, and the quantitative part, that is how much protein is present in the sample, that, as I have discussed, that you will prepare a standard curve based on the data that you obtain with respect to the bovine serum albumin estimation, and then compare your protein concentration, that is the concentration of the protein in your sample against this standard. And finally, the protein will be expressed in either milligram per gram or microgram per gram sample, whatever be the case. So that is in short about the estimation of the protein. And once more, if we just go through the basic reaction, that is reduction of Cu2 plus to Cu plus ions as a result of interaction with the peptide group of the protein of the analyzed samples under alkaline condition, copper cations form a complex compound with amide bonds of peptide groups during which they are reduced. The biuret chromophore is further stabilized by tartaric acid. Addition of Folin CO-Cal2 reagent increases the sensitivity of this method about 100 times. So you can understand that what is the mechanism of the blue color formation, okay, during the protein estimation. And that blue color is actually measured, the intensity of the color that you get, that is measured in the spectrophotometer. What is the wavelength at which you can measure this? It is normally 500 to 750 nanometer, where you can measure this blue violet color. And this is formed by the reduction of the phosphomolybdic phosphotungstic components in the Folin CO-Cal2 reagent by the amino acids tyrosine and tryptophan present in the protein. I think I have already repeated this. So that is the principle of the Lowry method. Again, you need to undergo some precautions. That is, you have to add, when you are isolating the protein, you have to add protease inhibitors. If the protein concentration of the sample is too high, you might have to dilute the sample, because if the color is too intense, then your reading may be out of range. So you have to dilute the sample and take the reading. Okay, you might have to take the reading again. You have to repeat maybe. A set of standards is needed with each group of estimation. Store the Folin CO-Cal2 reagent in amber bottles because it is light sensitive. Colored bottles, you have to store it. And they should have no greenish tinge. The protein estimation, if it is desired in a sample with high phenolic or pigment content, the extract should be prepared with a reducing agent like cysteine or NaCl. And all the standard curves should be prepared with utmost care. Because if there is a problem with the standard curve generation, then your the estimation of the protein in your sample, there may be an error in that. So you should be careful. So that is all about the quantitative estimation of protein, that is through Lowry method. Now finally, the last experiment which we are discussing today is the qualitative estimation of the protein. Like how will you measure the, as we have discussed, how will you measure the DNA quality? By running a gel. What is the gel? That is agarose gel electrophoresis. You are getting an intact band. So similarly, for protein, you have to again run one gel, and that gel is known as SDS PEG in short, that is sodium dodecyl sulfate polyacrylamide gel electrophoresis. We take a quick look about the principle of this experiment. Now for this experiment, what are the components that we need? First is polyacrylamide, next is bisacrylamide. Now what are the two main functions of this? Like in DNA, we are using agarose as a gel component. Here you are using polyacrylamide and bisacrylamide as gel components. So polyacrylamide are composed of polymerized acrylamide chains cross-linked by a bifunctional agent, that is methylene bisacrylamide. So you have to take both of these. If you need to prepare a polyacrylamide, you have to take polyacrylamide as well as methylene bisacrylamide. Now the concentration, again you have to 1:29. This is actually the recommended concentration, that is bisacrylamide is to acrylamide. The molar ratio should be 1:29. That is normally used to cast the gel. What is the function of bisacrylamide? It initiates the cross-linking. I mean, it strengthens the cross-linking of the polyacrylamide monomer chains. Then what you have to add? you have to dissociate the proteins. You have to denature the proteins. Before you are loading them in this gel, you have to denature the protein, and for that you have to use an anionic detergent, that is sodium dodecyl sulfate. Like in DNA, you have used a cationic detergent, that is CTAB. Here you are using an anionic detergent, that is SDS. And this SDS is used in combination with a reducing agent and heat. So SDS binding with denatured polypeptides, 1.4 gram of SDS per gram of polypeptide is proportional to the molecular weight of the polypeptides, which turn negatively charged. So this SDS, it has a particular stoichiometry in which it can bind to the protein. Then what you add is the ammonium persulfate, APS we call it in short, that is provide free radicals for driving the polymerization of acrylamide and bisacrylamide. And finally you have to add the TEMED, because TEMED, it accelerates the polymerization. This cross-linking or polymerization is facilitated by TEMED, but mind that you should add TEMED at the last part, at the end you should add TEMED, because once you add TEMED, immediately the cross-linking starts. So APS and TEMED should be added at the end, and immediately you have to pour the gel. You have to pour the gel. I will show the setup now, that where you have to pour the gel. Now for protein, first of all, when you are running a protein gel, it is not a horizontal, it is a vertical gel. In DNA it is horizontal gel, and here it is a vertical gel system. This is first point. And you normally use a discontinuous buffer system, because the pH of the buffer in the reservoir is different from that which is used to cast the gel. Now in case of protein gel, there are actually two components. One is the resolving gel and another is the stacking gel. That is SDS polypeptide complex loaded in the gel, they first migrate through a stacking gel. So stacking gel should be at the upper part and resolving gel should be at the lower part. Now the complexes accumulate in a very thin zone on the surface of resolving gel. This discontinuous buffer system allows concentrating all the complexes into a small volume, increasing the gel resolution. And the stacking gel, it has a pH of 6.8, tris chloride pH 6.8. The upper and lower buffer reservoirs contain tris glycine pH 8.3. Resolving gel contains tris Cl pH 8.8. So you can see three different pH systems are present. That is why we call it a discontinuous buffer system. The chloride ions in the sample and stacking gel form the leading edge of the moving boundary, while the glycine molecules constitute the trailing edge. And a zone of lower conductivity and steeper voltage gradient exists between the leading and trailing gel. This zone thrusts the polypeptide from the sample onto the surface of resolving gel. Because resolving gel, it has higher pH, it favors the ionization of glycine, and these glycine ions migrate through the stacked polypeptides and migrate through the resolving gel immediately behind the chloride ions. After getting free from the moving boundary, the SDS polypeptide complex migrate through the resolving gel in a zone of uniform voltage and pH and are separated according to the molecular weight. So the basic principle is that here the proteins are resolved or they get separated on the basis of their molecular weight, just like DNA. So the proteins which are of small molecular weight, they will migrate faster, they will be present at the lower part, and the proteins which are of high molecular weight, they will be present towards the upper part. But mind that the total proteins should be resolved in the resolving gel part, they should not remain in the stacking gel part. They should be all visible in the resolving gel. Now you stain the proteins with one stain, that is Kumasi Brilliant Blue. This is an amino tri-aryl methane dye that forms strong but not covalent complex with proteins via van der Waals forces and electrostatic interaction with the amino groups. It binds non-specifically to the proteins but not to the gel. And even if you have overstained the gel, there is a system by which you can counter stain it. Okay, you can de-stain it, okay, to remove the excess stain. That you can do. But for proteins, normally we use this CBB, we call it in short, that is Kumasi Brilliant Blue. So what are the materials required? The power supply. vertical electrophoresis system, gel casting plates and spacers, buffers as I already mentioned, acrylamide, SDS, ammonium persulfate, TAMED, tris-glycine electrophoresis buffer, protein molecular weight marker, protein samples, and the gel loading buffer. Because you need to mix the protein with the gel loading buffer which has SDS, because you need the denaturation, and it also has the stain, that is the bromophenol blue. So this is the composition of the resolving gel. There is a proper ratio in which you should mix the component: water, acrylamide, tris-chloride pH 8.8, SDS, ammonium persulfate, TAMED. Similarly for the stacking gel, 5% stacking gel, you have the mixture. You can find it in our self-learning materials. It's given there. And then you prepare the staining solution of, that is the Kumasi brilliant blue in methanol acetic acid, and then the destaining solution, that is only methanol and glacial acetic acid, not the Kumasi brilliant blue. So this is what the system look like, that is stacking gel and resolving gel. You have to load the sample, you have to fit the comb within the stacking gel, and then you have to load the samples as they have shown, and then you have to run the gel using the power supply, and then the proteins will be resolved. The big proteins will be in the, as I said, high molecular weight upper part and smaller proteins in the lower part. So I am not repeating the procedure because I have already mentioned that you have to prepare the gel, you have to mount the gel, pour the gel, okay, allow it to polymerize. First you have to pour the resolving gel, then you have to pour the stacking gel. Immediately you put the comb on the stacking gel, allow the gel to solidify, then load the protein samples. After mixing with the gel loading buffer, run the gel. Finally, you have to stain the gel in the Kumasi brilliant blue, stain it for 20 to 30 minutes, and then you have to destain the excess stain, okay, by putting it in methanol glacial acetic acid solution, and then you can observe the protein. Okay, what will be the observation? The proteins will look like this. Particularly if you see the protein in lane number 2 and lane number 3, you can see how the proteins are being resolved. Again, you get different bands. So Kumasi brilliant blue stained protein samples, and the first lane, it is showing the molecular weight marker. Again, the function of the molecular weight marker is enable you to understand what is the molecular weight of the protein of your interest. Okay, so here, since they have isolated the total protein, so they are getting, several protein fragments, because that in a system there are several proteins. So they are getting several protein bands against the molecular weight marker. Finally, before I conclude, again the precautions. So you should handle acrylamide, bisacrylamide with care, because these are neurotoxins. So you should always wear gloves. SDS, it is also a neurotoxin. So avoid ingesting, okay, and irritating, it might cause irritation for eyes and skin. Do not inhale the material. TAMED should be added at the end, because polymerization will start immediately. And when you are pouring the gel, this is another very important point. Make sure that you don't, no air bubbles are being trapped in the gel. Otherwise, they will interfere with the running process. Do not pre-run the gel before loading the sample, because this procedure will destroy the discontinuity of your buffer systems. So that is what I wanted to discuss for today. So we basically focused on 4 experiments: DNA isolation, okay, polymerase chain reaction, protein isolation, protein estimation, and detection of protein through electrophoresis, that is SDS page. So please go through this. These are very clearly given in the self-learning materials, lab course 2. Go through the experiments. I am sure you might have done some of the experiments also in your study centers. Try to understand the principle, basic principle. Okay, protocol to it's okay, you have done, but what are the principles? Why you get such observation? That is the important part. So go through the SLMs. If you face any problem with any of the experiments, you can contact us, write to us, email us. So we will be there to reply you and to help you in the best possible way. So thank you all, and please have a very happy experiments and a very, enjoyable experimental experience. Thank you all.